Good morning students, this is your teacher Neelam Elawat and today we'll start with the new topic that is referred to as translation. Now before we start this process, we must understand the terminology that we have used in the last video. Okay, let's recapitulate what we have done. We know students that DNA can produce DNA. Okay, DNA can be produced by DNA and that process is called replication. And from DNA, mRNA will be produced, right? This mRNA will be produced and this process is called transcription. This process is called transcription. We have talked about it in detail. And now from this mRNA, proteins will be made. The process of making proteins, the process of making amino acids from the mRNA is called or is named as translation. This process is named as translation. Okay. Now before we talk about the translation, there are certain components that are needed for this translation process to start. Very first of them is the ribosomal units. First, the two ribosomal units, one is a bigger one and second is a smaller one, they will unite to form a complex which is called made up of two ribosomal units, and only then this ribosomal complex will be active. If these two units, if they are separate, they will remain inactive. They can function only when they are united with each other. They are combined with each other. Secondly we'll require the mRNA. Okay, we will require the mRNA because mRNA has the information, okay, and from which we can transcribe the proteins. Third is the tRNA. The information which is present on mRNA can only be read by the tRNA. The information which is present on mRNA is in the form of codons. Okay, I hope you remember the genetic code. The genetic code is always in the form of codons. So the mRNA has the information of proteins or amino acids in the form of codons, and these codons, they could be read by the anticodons which are present on the tRNA. Right everyone? So we require basically three things for the translation process to start. First, combination of ribosomal subunits. Secondly, the mRNA, and thirdly, the tRNA. Okay, the information for reading the tRNA for reading the information which is present on mRNA. Now coming to the process of translation. This process we can divide into 5 basic steps. First is the activation of amino acids. Second is attachment of mRNA and the formation of initiation complex. Third is initiation of polypeptide chain, elongation of polypeptide, and the last is termination. Okay. Now let's discuss each step separately. Now very first is the activation of amino acids and the tRNA. This process is also called as charging of tRNA. Right? This process is also called as charging of tRNA. Or we can call it as amino acylation of tRNA. Now this process, charging of tRNA, is done by the amino acyl tRNA synthetase. And for this process, magnesium ions are required. So this process is very important and can only occur in the presence of magnesium. Basically what it happens, the tRNA combines with amino acids. So this is your first step. The tRNA combines with the amino acids and thus it is referred to as charging of tRNA. Okay, amino acids become attached to the tRNA and this step is called charging of tRNA, and this attachment is done by the enzyme amino acyl tRNA synthetase, and this combination can only occur in the presence of magnesium ions. So let's see how it happens. So this is you can see this is your tRNA molecule. The tRNA molecule becomes attached with the amino acids. Here the amino acid is leucine. Okay, this is the first step, right? Is it clear to everyone? So first of all, this has occurred. So tRNA becomes associated with the amino acids. Now this tRNA, it becomes charged. Okay. Now always the initiation process will start. The initiation process will start with AUG codon. Okay. It will always start with the AUG codon. This is your mRNA and this is your codon AUG, which is called your initiation codon. And this will pair with the anticodons present on the tRNA. And we will have amino acid methionine here. So methionine will be formed. Okay, dear students, which compound will be formed? Which amino acid will be formed? Methionine will be formed. Okay, clear? So first the tRNA, bounded tRNA, which is bound with the amino acid, will bind with the initiation codon or the start codon, and that initiation codon is AUG, and it will automatically code for the methionine amino acid. Okay, dear students? So attachment of mRNA and the formation of initiation complex, right? Just now we have discussed it. We've discussed it just now, right? This is exactly what has happened. Okay, your initiation codon will bind with the anticodon present on the tRNA, and to this it will bind the amino acids. So we will start with the process with start codon. So mRNA will attach with the ribosomal unit. mRNA will attach with the ribosomal unit at the start codon. Okay, and start codon will code for methionine, right? Will code for methionine, and this complex which is formed is called initiation complex. This complex which is formed is called initiation complex. Okay, dear students, what do we call this complex? Initiation complex. Okay, clear everyone? Is it clear? So this complex is called your initiation complex. Okay. Now tRNA, which is bound with amino acid methionine, is also called as initiator tRNA. So what is an initiator tRNA? The tRNA which is bound with amino acid methionine, right? This will be called as initiator tRNA because this will start the process. Okay, this will start the process. Now this position which is here, you can bound it, it's called a P site, right? This position is called a P site. Okay, dear students? Now next step is initiation of polypeptide. Okay, it is called initiation of polypeptide. See, now one amino acid which is methionine has been formed. Okay, first amino acid is methionine. Now next amino acid will be formed in series and they will be attached with each other. So it will result in the formation of a long chain. Okay, and that chain is called a polypeptide, right? That chain is called polypeptide. So the methionine binds with P site and the next amino acid or the next tRNA will bind with A site. Okay, let's see the P and A site. So this is your A site. Okay, dear students, and this is your P site and this is your A site. Okay, your initiation codon is bound to the P site. You must remember it. It is many times it is being asked in the exam that the initiated tRNA will bind with which site? It will bind with P site. So your initiation codon is AUG and this tRNA is called initiator tRNA. So second amino acid will come and bind with next codon, right? Second will come and it will bind with the next codon. Okay, this second tRNA which has come is having the amino acid leucine. Okay, this is having the amino acid leucine. Now there will be a formation of bond between the methionine and leucine. Okay, methionine and leucine, they will be interconnected with each other. They will become connected with each other with the help of a peptide bond, right? With the help of the peptide bond, right? With the help of the peptide bond. Thus it is called formation of polypeptide. So slowly and gradually the next amino acids will keep on adding like this and this will form a chain. So elongation of polypeptide. As I said, that the amino acids, they will keep on coming, they will be joining with each other, right? They will be joining with each other one after the another. So this is called elongation of polypeptide. So elongation always starts from 5' end and it reaches to 3' end. The second tRNA molecule will always bind with A site. Okay, dear students, the second tRNA molecule will bind and come to the A site, right? Then there is a formation of bond between the amino acid which is present on the P site and the amino acid which is present on the A site, and this bond which is formed between the two amino acids is called a peptide bond, right? This is called peptide bond. Clear? Okay. Now what basically happens? See here, the second amino acid, second tRNA, it is present at A site. Yes, it is present at A site. Now slowly what will happen? This will shift to this position. Let's see. Okay, the tRNA which is present at the A site, it will then shift to P site, and this is called translocation. Okay, in first position, our methionine tRNA will attach with the P site and the second will attach with A site. Now P site will become empty, okay, because methionine amino acid has been formed. Okay, now tRNA will leave the place. Okay, now the tRNA which is present on the A site will come to P site, and the new third tRNA will attach with the A site. Okay, dear students, clear? So first P and A, methionine was attached at P and second was attached with A. Now this methionine will go. Okay, methionine has left, P is empty. So A will shift to P and a third tRNA will come to A. Okay, and a third tRNA will come to A. Let's see this. Now see, at this position, your P site has become empty. Okay, P site has become empty. And just imagine, here it was methionine was present. Now see, where is methionine? The methionine is attached with leucine. Okay, so both the amino acids, they are attached with each other and the bond between them is called a peptide bond. So now a P site is empty. A P site is empty. Okay, now this will move, this mRNA will move further. Okay, and this tRNA will shift to P site. Then the third tRNA will come to the A site. Okay, clear everyone? आपने देखा होगा when you are standing on the ticket counter, होता है ऐसा, आप ticket counter पर खड़े हैं, so the person who is in front of you takes the ticket and moves away. So you move a little bit closer to the window, isn't it? So same way, this person has taken the ticket and has left. Now he has entered into the movie. Now you will come to the ticket counter and will take the ticket. Okay, clear everyone? So this second tRNA will shift to the P site and third tRNA will come on A site. So in this manner, you will have attachment of amino acids. Okay, so this has left. Now this has come to the P site, second tRNA has come to the P site. Simultaneously, this mRNA is also shifting. ठीक है, mRNA भी shift हो रहा है. Clear? Is it clear to everyone? So now this information will be read and leucine will be formed. Now the two amino acids which are present on T will be shifted to the third, which is glycine. ठीक है, amino acids को यहाँ shift कर दिया जाएगा and this tRNA will be free and it will leave. Clear? Is it clear? So you see the ribosome complex is continuously shifting its position on the mRNA. ठीक है, पहले यहाँ पर ribosomal unit मौजूद थी. Now it has shifted one position, it has shifted one code, so that the second tRNA is present at P site and the third tRNA is present at the A site. Okay, now fourth tRNA has also come. Now once this finishes its work, it will leave. This ribosomal unit will again move. It will move in this particular direction, and this is your 5', 3' direction. So ribosomal unit is continuously moving from 5 prime to 3 prime direction. Now again see, in the same manner, 1, 2, 3, 4. The 4 amino acids have been formed. The very first is methionine, second is leucine, third is glycine, phenylalanine, and now again methionine. So in this manner, the ribosomal unit is continuously moving along the mRNA, right? It is continuously moving along the mRNA, and the amino acids, they are being formed, and amino acids, they are being joined with each other, resulting in formation of a long chain, and this chain is called a polypeptide. Okay, this chain is called a polypeptide. So amino acids, they are joined with each other, they are forming a chain, and that is why it is called a polypeptide. Now see, the mRNAs, this ribosomal unit is shifting from one place to another, and now this is your stop codon, isn't it? It is reaching the stop codon, and we have got a chain of amino acids. Okay, we have got a chain of amino acids again. So we'll reach the termination process. Now 3 codons, they will be acting as the terminator, tRNA terminator. Okay, they will act as terminator, or they will act as stop codons, means they will stop the process. And this stop codon, it will be always on A site. Okay, it will always be on A site. Clear? Now tRNA cannot identify the stop codon. Okay, generally we have seen that what happens, the codon is read by the tRNA. tRNA comes, joins with it, and it starts the process. But the tRNA cannot identify the stop codons. Okay, so the tRNA which is present in the P site, that is released, okay, in the presence of certain releasing factors, and the next tRNA is not able to bind with the A site, and thus this process will ultimately stop. There will be a dissociation process that will occur. The two ribosomal units, they will separate. Okay, they will separate. See, earlier mRNA and the ribosomal units, they had combined. Now these two units, they will become separate from each other. The tRNA will not be able to associate, and we have a newly formed protein. So protein is nothing but it is a chain of amino acids. So what is a protein? A chain of amino acids. Now the initiator complex, right, that was assembled, will now separate. Each component will separate, will dissociate, right, and once the ribosomal units, they separate, they will not be active now, right, and this is done by whom? The stop codons. The stop codons which are present at A site, they are not identified by the tRNA. So thus this is called dissociation of the complex. This is called dissociation of the complex. Okay, I hope you've understood. So coming to your homework for today, first of all, make notes of the topic and beautifully draw the diagrams, because through diagrams you can easily understand the process. So beautifully you draw the diagrams, right? Secondly, you read the page number 114 to 115, underline the important points, and if there is any query, any doubt, you are most welcome. Okay, you're free to ask me. Okay, dear students. Okay, so that's all from my side for today. Goodbye, everyone.